thank you all for coming here for this start of our afternoon sessions. And we're going to take a different turn a little bit here because we're going to be talking about some more in the biomedical side. So we went from robotics to biomedical. And I'm going to have our speakers introduce themselves. Please, is your mic, our mic's working? You should be able to turn it up and then it turns green. And then I always tap it. Okay, great. Take it away.
Okay, hi. Thank you for the opportunity. I have been in conversations with Dr. French since before COVID, I think.
Yes. Yeah.
It's like instead of BC and AD, it's like pre-COVID and post-COVID. So I'm Anirban Sengupta. I'm a faculty in biomedical engineering at Case Western. I have been a faculty for ever now, 22 years. I had to count it out. So we do a lot of work broadly in the blood space. And obviously one of the big areas in the blood space is blood transfusion. So one of the biggest killers globally, and this is not just a military thing, this is a civilian military combined thing. One of the biggest killers is people just bleeding out from accidental injuries. So think of car accidents, think of people, falling off the roof, trying to hang their Christmas lights. It's obvious to understand injuries in a war zone. And then unfortunately, there is a lot of terrorist mass casualty related events that happen. In all
active shooters.
Yeah, active shooters. And I mean, these are unfortunate but real scenarios. None of these are diseases. What kills people here is just bleeding out, because blood is our sort of lifeblood, so to say. Without blood, we don't survive. It is carrying our gases, our nutritions, everything else to keep ourselves alive. And so if we lose a lot of blood, we die. And the only treatment for that is to give blood back, which is called blood transfusion. Unfortunately, the only source of blood to give blood back is donors. And you might have seen American Red Cross blood drives, things like events that happen in your neighborhood. Those are all geared towards getting donors to give blood so that we can have blood for a certain period in stock in case we need it, but there aren't ever enough donors. And then when you get scenarios like the pandemic, COVID, blood drives go down even further, and then national emergencies get declared because of, dearth of blood, because cancer patients need blood transfusion. Other people who are born with blood disorders need blood transfusion. It's not just injuries. And obviously, the military side of that is, always having blood in stock in case there is a war, in case there are battlefield injuries, and in case the military need transfusion. Biggest problem is, again, never enough donors. And then blood is a biologic product, so it can only be stored for limited periods of time. It cannot be stockpiled. So one of the areas that our lab does research on, it's currently funded by DARPA, which is a military entity, is to make fully artificial blood, something that works like blood but doesn't need donors, and something that can be made into a powder and stored for long periods of times, which is stockpiled theoretically indefinitely, and then all you need to do is add sterile water to it as you need it and use it on demand. So that's the big project that Dr. French and I are involved in. Dr. French is in charge of the ELSI oversight of the project. I, as well as multiple people at Case Western and at other universities, collaboratively are involved in building the technology of the artificial blood. And that's the context in which I have the opportunity to talk a little bit about but I can talk about other research too and other things too that kind of fits into the mold of the ELSI philosophy. So thank you again for the opportunity, and happy to be here.
Wonderful, and it's an honor to work with you as well. Lynette, please tell the audience about yourself.
Hello, everyone. It is my pleasure to be here. I am an assistant professor in the Department of Bioethics. In case you're wondering about my funny last name, it's pronounced Jerido. So I have a PhD, and I did an ELSI postdoc at the University of Michigan. I focused more on the human genome and things like that. But I'm also trained as a software engineer, and I've worked in a past life many years in industry and talking about tech. So I like to look at these biotechnologies. And so thank you so much for giving us this overview of some of the work that you've done. And I have some questions, just to kind of get things started. And you talked a little bit about artificial blood technologies and, gave us some insights on how they might be used. Do you happen to, could you give us some kind of milestones or information about how it's evolved over the years? Because it's not necessarily a new technology, correct?
Sure. Yeah. No, this is a great question, and it has a very rich history. The history actually goes back to not military use, but another global pandemic called HIV, AIDS. So, a big area of artificial blood or bioartificial blood research evolved because of HIV and due to the risk of transfusion-associated infection, HIV. So in the 80s is when the HIV risk scenario was recognized from transfusion framework. And a lot of folks, a lot within the US, but outside the US too, in Europe and in Japan, started working on, an alternative. They wouldn't call it necessarily artificial blood,
Right.
because that's a loaded word, but an alternative for transfusion that is the phrase that people try to use, that is donor independent, which means it can be manufactured in a lab. It's donor independent, okay? But I would say the lion's share of that research focused on a single entity within the blood space, which is oxygen carrying. So if we think of blood as a complex fluid, there are some cells in it, and there are some molecules in it, and then there are some ions in it. So it's like a mishmash of things. If we look at the cellular part of blood, one big part of that is this cell called red blood cell or erythrocyte. And that's why blood is red, because there's a protein inside these red blood cells called hemoglobin, which in the oxygenated form is red in color, okay? And so hemoglobin is the protein that binds oxygen, that allows us to breathe and then load up our red blood cells with oxygen. And then as the blood flows into the tissue, it downloads the oxygen to the tissues and the cells to keep them alive. And then as blood comes back to our lung, it lets go of the carbon dioxide that we expire out, and it leaves our body. So this function of tissue oxygenation is a critical component of blood. And that's the part where majority of the research focused on, both in a university setting, but big pharma. So Baxter was involved in it. There was a company called Northfield Corporation that was involved in it. Alliance Medical that was involved in it. Johnson & Johnson was involved in it. CSL Behring, which is again another big pharma company, were involved in it. And a variety of products got developed in the context of what we would call oxygen-carrying artificial components or artificial cells. But unfortunately, all of them failed clinical trials. So they were all great from a scientific design standpoint. And then they did kind of okay in, for example, animal model studies and things like that. And that has an ethical aspect to it by itself. But as they moved into clinical trials, which means testing in humans, some of them went from the safety clinical trials, which we call phase 0, phase 1 trial, to efficacy, does it actually carry oxygen or not, to an injured tissue, which is phase 2, phase 3, and ended up failing in the phase 2, phase 3 scenario, causing risks of stroke and blood clots in different scenarios. Looking back in retrospect, we understand why it happened. There were certain scientific rigors of testing that was missing even before going to clinical trials. And hemoglobin, the protein that carries oxygen, is a potent clot promoting protein. And people didn't think it through, and there was like free hemoglobin floating around. So you were, basically always at a clotting risk. So as it failed, in 2006, a position paper, a review article, was published comparing all the different hemoglobin-based oxygen carriers that were developed through 2 decades, and concluded that this is not a safe approach. And that kind of really put a death nail in the field of doing artificial blood. Somehow, in the background, the need never went away. So somehow in the background, this, what if we could make it happen? Now that we understand the science a little bit more. That question remained. But to make it happen means starting from zero again, because a whole field kind of died down over 2 decades. And it required a lot of financial support. It also required an entity that could call the shots without necessarily getting embroiled in politics and academic barriers and things like that. And the only military entity that can do this is DARPA. So DARPA is sort of, DARPA came into existence out of surprise when Russians launched the Sputnik. And US went, oh crap, what just happened, right? And so if you think of I think I walked into a conversation about tech excellence. US was left kind of stunned, oh, we didn't even know about Sputnik. What just happened, right? And that prompted creation of this organization called Defense Advanced Research Program Award, which is focused on tech excellence, not just medical, but it has, cybernetics, cybersecurity, weapons, telecom, satellites, whatever, and medical. They are currently building gadgets to snatch scrap metal from destroyed satellites in the sky and bring them back to the Earth to recycle metal. So they do these kinds of blue sky projects, right? And so DARPA recognized artificial blood as a blue sky project because it has been attempted for 2-3 decades and did not succeed. And so they talked with, including myself, a variety of people that have been involved in this area of research and came to the decision that no, they should invest in this, and this is a worthwhile investment, not just for the military, but for a global need. Already blood availability is a problem in the US, the most developed nation in the world. So think about nations that are, developing nations. I come from India originally. Blood availability, my dad is a cancer survivor. They had to go out asking random people in the hospital to give blood to give him transfusion. Blood vigilance is not a big thing yet in developing nations. So if an artificial blood gets developed, it not only helps the military, but it goes out back into the world to help scenarios where donor-derived blood is not easily available. So this kind of thought process brought back the concept, but it also brought back the recognition that blood is not just red blood cells. Red blood cells do one part of it. There are other parts of blood that do other functions that are equally important. For example, blood contains something called plasma, which is the liquid volume part of blood, but that plasma is responsible for maintaining the blood flow at a certain speed, maintaining the temperature in our body, carrying red blood cells and other things to wherever they are needed. So you need a fluid system, which is the plasma. And then if there is an injury, our body has a very elegant system to try to quickly form a clot to prevent us from bleeding out. And that is driven by a kind of blood cell called platelets. So now when DARPA came back to make artificial blood, they paid attention to all the different components of blood, and then they talked with investors. So I work in the platelet space myself. So they talked to all the investigators that do different parts of this blood research, and then created a program to bring all of them together under a pretty heavy funding support, like almost $50 million, with the goal to get to the point of a successful artificial blood in 4 years. And that's the kind of thing that DARPA does, that, all right, we're going to give you all the money you need, but you give me the product tomorrow, right? So this is the philosophy that they run by. And so we are in year 3 of the endeavor. And this is where now the blood endeavor has come back. And this consortium across multiple universities is the only consortium in the whole world that's actually developing a bioartificial blood. So it's really a humbling experience to be part of this team and figure out how to solve this.
Thank you so much for that. That's really rich and helps us to understand, what's happening beyond the oxygen delivery models. Could you talk a little bit more about how your approach to synthetic blood components and research is kind of different than before? Obviously, you're mentioning that, but just maybe some of those lessons learned in how you've developed, how this consortium operates and kind of shares information and things like that, and how far you may or may not be to clinical trials.
Sure. Well, the clinical trial part I can't answer right now because it's TBD. Oh, the clinical trial part I cannot answer right now because it's TBD. We'll know in 2 years.
Okay.
But I'll cycle back to the clinical trial, or like what is needed to get to the clinical trial, because there's a pretty interesting path to get there. So if I go back to the history, and I was kind of mentioning, this sort of 2 decades or almost 3 decades of research in artificial blood that failed, all of those attempts were utilizing this oxygen-carrying protein, which I named hemoglobin. But they were just taking hemoglobin out of either expired blood or hemoglobin out of cow blood, like not human source, but other animal source blood. But taking the actual purified protein and then trying to make a product with the protein itself. You cannot really fool nature, okay? There's a reason that 1 million or more than 1 million years of evolution has kind of designed things to work in a certain way in our body. In our body, hemoglobin doesn't float around openly by itself. It's encapsulated inside a cell. So it's not open. And the reason for that is evolution or nature has designed it in such a way so that there's a protection barrier around the hemoglobin so that it doesn't get exposed to things that it shouldn't get exposed to, okay? So all the designs that failed clinical trials did not really pay attention to that. So what we are doing now is we are still using hemoglobin for the red blood cell mimicking or artificial red blood cell design, but it's loaded inside a particle that has a shell with a certain protective barrier that doesn't expose the hemoglobin to the outside environment of the blood, which I'm going to skip the details, but which actually prevents all this clot risk things that I was mentioning that hemoglobin does. But the shell is designed in such a way so that it, just like real red blood cells, helps oxygen enter and bind to the hemoglobin inside the particle in the lung. And then when the particle goes to the tissue, it actually releases the oxygen, and the oxygen crosses the shell to go into the tissue. And this switch between loading and offloading is maintained by pH, the acidity of the shell, because our lung acidic environment is very different from our tissue acidic environment. So using pH as a trigger or a switch, it can upload and download oxygen. So this is one difference. Platelets, on the other hand, they're like a Swiss Army knife. They have a variety of different functions that need to work in concert for them to actually form a necessary clot, but not form an unnecessary clot. And so what my lab has figured out is how to control this thing by, again, using synthetic particles that have the Swiss Army knife functionalities, but those functionalities become active when needed. When it's not needed, they are not active. So it switches on or switches off, depending upon the need. And then if I can bring it back to plasma, this is a funny story. So during World War II, so going back to military technology, during World War II, the French had figured out how to freeze dry plasma into a powder. They had designed machines to do that. For some reason, the US left the machines in France, never brought it back on US soil. So for decades, it was only the French that was producing this plasma powder where you can just add water and it becomes plasma. And during this DARPA endeavor, people realized, like, what the heck are we doing? This is, I mean, we kind of designed this thing. We should use this ourselves. So there are a number of US companies now. So one of them is a company called Teleflex. One of them is a company called Velico. So these are all US-based companies, but they obviously have global presence, so in Ireland and certain European nations, that have now designed processes and machinery to freeze dry plasma. And the idea here is that the artificial red blood cell would be a freeze-dried powder. The artificial platelets, we have already made it into a freeze-dried powder. And then the artificial, or not artificial, it's biological, but plasma has a big, pretty long shelf life. So the plasma will be a freeze-dried powder. And you can store these things as powders for long periods of time without the need of donors, because these are just lab-made stuff. And then as needed, just add water. And now you have your artificial blood. So this is a big difference from how the so-called blood mimicry was envisioned back in the 80s and the 90s and early 2000s. So that's the big difference.
Yeah, thank you for that. That's a great way of kind of distinguishing and showing how, you know, we've learned from the past and also opportunities for the future. One, because, you know, there's a lot of conversation about, okay, how do you have access to this thing? We're hopeful that you get to the, you know, over the finish line and get to the trials.
Oh yeah, I forgot touching up on that. Yes.
But you know, one of the things that, you know, I think it's great is that you're in this phase before the trials and some of the considerations ethically that you're taking into account as you're developing and planning for a future where you are going to be testing. How do you think you would approach, are you doing anything differently in your study design now to prepare for when you start doing human subjects trials?
Sure. So let me answer that question by backtracking what's needed for human trials and hence, you know, what's needed now. Right? So this would be the artificial blood, or I should say the artificial blood components even individually as standalone, would be considered therapeutics. These are drugs, right? So these are therapeutic compounds. To use a new therapeutic compound in humans, and hence to first run trials in humans, FDA requires something called investigational new drug approval, IND approval. And this IND approval, so this kind of connects to, you know, regulatory path, but it also connects to the ethical aspects of it. To do IND, so now I'm going to backtrack. To do IND, first there is a need, unfortunately, to do, according to the FDA, this is not my rule, according to the FDA, there is a need to do a certain limited set of animal model studies. This is unavoidable in this kind of drugs. So for certain kinds of drugs, you can do what we call in vitro, like lab bench studies and not in a live animal. and it's fine. FDA will accept the data. But something like blood transfusion, they require certain animal model studies. Now, if I think of the animal model studies, here comes ethical concerns, because the animal model study itself is an ethical dilemma, right? I'm not a big proponent of it, but there is no way around it, okay? So if there is no way around it, the next question becomes, what animals, right? Now, I'm kind of assuming here, but I'm pretty sure in our lifetime, most of us have killed a mouse somewhere, in the house, in the field, whatever, run over something, whatnot, right? And we don't feel a lot of, oh my God, I killed a mouse, when we kill a mouse. But if you go up the scale of the species, which is what is required to get it closer to the anatomy of a human being, a lot of complications start emerging, right? Now, FDA, again, not my rule, this is the FDA rule, they require safety studies, not efficacy means it works in an injured scenario. Safety means if you inject it into a person, it will not kill the person. You have not made a lethal injection, right? So that safety thing needs to be tested in animals first, and FDA requires 2 different species of animals to be tested for safety, and then escalating doses, various doses. A small dose may not kill you, a higher dose may kill you. So they want to know what is the limit beyond which you, or which you should not cross if you are to ever give this to a human. So, the small animal species in FDA's eyes, if you choose a mouse or a rat, it's okay. People don't really, get hung up on somehow, which is surprising to me. It's still an animal, but don't get hung up on, oh my God, you're gonna inject a mouse, you're gonna inject a rat. They don't get hung up on that. They do get hung up if you say, I'm gonna inject a dog, I'm gonna inject a rabbit, I'm gonna inject a monkey, a sheep, because these are all in FDA's guidelines, large animal species, but it is required currently to do that. So this is an ethical dilemma that we all try to navigate, and our only hope is minimize. We can't avoid, but minimize. So there's something we do in statistics called power calculation, which is, you determine a certain level of effect that you're hoping to see with a certain amount of confidence that every time I'll do this, every time I turn the switch on, the light will turn on, right? You can't ever say that with 100% confidence if the circuitry is bad, but you can say that with a certain level of confidence, right? So you could do, there are mathematical formulas for that, so you could do effect size based confidence associated calculation of how many animals you may need to convincingly prove that yes, the stuff works. It is going to be safe. And that's the approach that we have taken. Our animal models are, unfortunately, rabbits and non-human primates, monkeys. Okay, because the monkey anatomy is the closest to the humans. Now, we can all make dire statements about Planet of the Apes and all this kind of stuff, but non-human primate research is there, has always been there. What has happened is the rigor in which it is controlled, and the heavy focus on people not like messing around with it. Like, if you can calculate 7 monkeys is all you need, that's all you get. You can't say, oh, just give me 10 more, just in case, kind of thing. So that's a rigor, right? So now I'm going to go back to the clinical trial side. So let's say it passes all of this, and it gets to the point of clinical trials. The first trial, just like we are having to do or going to do in animal models, the first trial is always safety, which means you take healthy volunteers, humans, healthy volunteers, and you inject what you think is the therapeutically appropriate dose of that system, in this case, the artificial blood, and you basically observe for what we call adverse events. And the adverse events can range from little fever to dying of stroke. Like, those are the two extremes. Little fever, we are okay with. We all got vaccinated. We kind of felt yucky for a day or two, and then we were fine. So that's an adverse event, but that's a tolerable adverse event. FDA is okay with that. But if I had a stroke, just like some vaccinated people, due to a very specific phenotype that they have in their body, they got something called VITT, V-I-T-T, which is vaccine-associated clotting, and some of them died. Very rare cases, 1%, 2%, but it does happen. So you want to know that, and you want to know if that is happening, what's the mechanism behind it, so that you can pre-screen people from getting that, right? So that's the first safety clinical trial. Once that passes, then it goes into something called efficacy trial, which means now this would be used in patients that are requiring blood transfusion because of a situation. So think of complex surgeries. Think of cancer patients whose bone marrow is shot because of chemotherapy. These are the kinds of scenarios where the efficacy is evaluated. And then if the efficacy trial is successful, then FDA provides an approval, and then it can, what we call, expand into other parallel applications. So maybe it gets approved in cardiac surgery, but it can then be used in hip surgery or cancer or whatnot. Now I'm going to touch upon a parallel but different thing in the military context, because we're talking about certain military applications. Military has something called EUA, Emergency Use Authorization, which is a very different context than this clinical trial path that I talked about. An emergency use authorization involves recognition of a scenario where the technology is in such a dire need that you don't have to go through all these clinical trials. As long as you can show, let's say, safety, they're going to take it to the field and give it to soldiers. Okay? So that's a different path. And that kind of EUA only happens in extreme situations. What's an extreme situation? I'll give you the most prominent example right now is COVID. COVID had an EUA for vaccines and convalescent plasma to save lives of people that were dying by the millions. So that's sort of the context.
I'm not cutting you off. I just want to let you know we have 10 minutes. So if we have audience questions, we want to leave a little space for that as well. But Lynette, if you want to comment on what, or just want to open the floor.
No, I was going to
open the floor. Okay.
All
right. So please use our mic. You see there's a mic in the aisle there. If anyone has a question, so that we can all hear, because it's a bigger room than it looks in terms of the length and can't always hear.
Are there any human blood products in your artificial blood that would cause problems with prions, COVID, HIV, Lyme disease?
Yeah.
Are the three that come to mind. So are there any blood products in the artificial product that you have to screen donors for or screen your supply for before you make it?
Yeah, so we are yes. So the first answer is yes, because the hemoglobin is coming from expired red blood cells, which are from human donors. So instead of throwing away the red blood cells, people are just extracting hemoglobin out of it and reusing it. The plasma part is also human derived, because these are human donors. You must have seen plasma centers, and that's the easiest blood donation is the plasma center. But instead of pre-screening, the U.S. has the world now, but including the U.S. Has developed something called pathogen reduction technologies, PRTs, and so all of these products undergo this PRT treatment to deactivate any and every risk before it gets into use. So in the current artificial blood endeavor that we are talking about, the only fully artificial, not human derived component is our platelets. That's just fully synthetic. But the other two components have some human derived aspects. So the final product would be a PRT product such that all these risks are minimized. But great question. Thank you.
Oh, that is a great question.
Nobody else
Well-
has another. Do you have an economic differential yet as to what the plasma industrial complex does in all of our urban neighborhoods versus, I mean, are you still using them and still need them in this process to make it work?
Yeah. So plasma donation is way more common than blood, but it is not sufficient ever. So it's interesting that you asked that, because our proposal to DARPA was that we're going to make an artificial plasma too. Because plasma is basically a mixture of proteins, and you can make proteins now with something called recombinant technology. So you can make proteins. And we proposed to them that we will make a fully artificial plasma, but that proposal, including all the other stuff, was much beyond what they could afford in their budget. So we are unfortunately still dependent on donated plasma, and the financial boundary condition that Pentagon has put on us is that a unit of artificial blood cannot cost more than a unit of donated blood. That's the only thing we have. So currently a unit of donated blood, what we call fully loaded cost, which means the cost of the donation and the cost of the process and the cost of the transport. And if you take all of these into consideration, the logistics into consideration, a unit of donated blood now costs about $2,000
Wow.
a unit. Our artificial blood should not cross that number. That's the only requirement for us currently.
Now, I'm going to pick on our our visitors once again. All right, I'm going to hand you.
No, I don't need a mic.
Okay, you can.
Yeah.
Well,
they
it's
can't
not
hear
for
you
you.
in
They
the
can't hear you in the back. I'm afraid.
Here, I can hand one over. I can take that mic.
Oh,
There you
no,
go.
I'm sorry.
No, it's all good.
Here you go. Here's a different.
I can
one.
scream.
I don't know if you already covered this or not, but how would you get over the barrier of like different sorts of blood types? Like, is there a way, like, would you just use like a universal for every single one?
I mean, this. Fantastic question. So the beauty of putting hemoglobin inside a synthetic particle is that the particle has no antigen. So the product, by default, is what we call immunosilent. There is no antigen.
Wow.
So it's a universal donor no matter what. Okay? So the antigens in real blood cells are on the surface of the cells. Okay? But if you put the protein, that's the machinery of the cell, but inside a synthetic particle whose surface doesn't have any antigens, the product by default is immunosilent. So there is no donor mismatch by default. So which is, I'm actually super impressed that you asked that question, because people don't think of this very often, and I didn't want to get into the nitty-gritty of that, but that's an awesome question. But yeah, it's universally applicable to not just humans, any species, because people are thinking about going back to economics, a veterinary medicine world of it, right? There is no blood bank for dogs and blood bank for cats, but there are certain species of dogs and cats that have either congenital bleeding problems or might get run over by a car. How do you save them, right? At Ohio State University, they have a bay where there are donor dogs and recipient dogs getting body transfused from each other. But think of artificial blood that is species agnostic and type match agnostic. You can give it to anybody if it's successful.
And feel free to jump in. I was just going to say,
wanted
Lynette,
ask
jump on
one
in.
question that kind of ties some of these topics together in terms of ELSI. There's opportunities, like you mentioned, there's a lot of plasma donation and things like that in different communities. Is there ever any kind of engagement or outreach to say, your blood is being used for research, and this is a promise of tomorrow in a very hopeful way, because like what you're describing is transforming this to like a universal donor, donor independent model that could be very helpful and save many lives, and just as an encouraging opportunity.
Yeah, so not at this stage, because this is still in the preclinical stage. I think the advocacy is the word that I would use, and sort of the communication and advocacy.
Right.
I think that usually would start once it's IND approved and will go into clinical trials, because then there's a possibility that it will become a product.
Okay.
So then there is a conscious effort to educate globally about both the utility of, if they are donating plasma for this, or the utility of using this.
That's right.
And then there are, for the lack of a better word, there are target communities like Jehovah's Witnesses is an example. Jehovah's Witnesses are religiously prohibited from taking biologically derived transfusions. So they don't take human transfusions. There's a Jehovah's Witness center up on Cedar, I think. That doesn't mean Jehovah's Witnesses don't get into car accidents or get cancer. So they need blood transfusions too. Now they will have an option, but they don't know that this option exists, because we haven't gone to that community and told them that, hey, this is happening. So all of those would start sort of, probably in 2 years' time, if we get to that IND phase.
Which we hope we do.
Yeah. This will be fantastic.
Other questions? We have Nick over here. Jake, you run that. Oh, here, she can.
I'm good.
You're good.
Is
Okay.
Is this on? Okay.
Sorry.
So I think maybe this is a question for the whole panel up there. It seems like with biotech, the ELSI parameters are baked into the FDA process to a, not entirely, but to a pretty good extent. So it got me thinking, like, when I think about information technology or AI, these types of things that may not have direct biological effects, but may have psychological effects on the community, like, why don't we have something similar for those types of processes to go through? Reflections on that?
I mean, I'd love that, obviously. I don't know, Lynette, why do you think we haven't managed that yet?
I mean, I think that we were, and internationally, we were moving in that direction. Like, there are treaties for, the ethical use of AI.
True.
And there are a lot of consortium that are doing that. I think that we have that push-pull tension that we have with most technologies, where there's we don't want to kind of rein it in too soon and to challenge innovation. That's the ideas of some people. But at the same time, I think that there's opportunities, like were discussed here, to learn from past technologies and challenges that we face to improve our models of research and delivery. The other thing is, I think that because, like you mentioned, it's not something as physically and visibly in front of your face, this kind of challenge that someone is potentially bleeding out, or it's not a specific disease or condition, that they're still working to link some of the things that you might translate to AI, the use of AI and social media and things like that we were discussing this morning. So I think that those connections, even though we're studying them and seeing the impact, we're still doing that in real time. And so I think, like many technologies, we're kind of in that area where it is not heavily regulated, because we're still trying to figure out what are the outcomes of the impact of this.
Right. And something to add to that is, I think the data is still kind of emerging regarding what we may consider good use versus bad use of AI. So I'll give you, in the medical world, I can give you multiple use. I mean, AI is actually connected to the biomedical world. For example, drug design, right? The traditional way of drug design is run 10,000 experiments, see which works, and then identify a candidate. All these experiments work on structures of molecules. And so they'll have 10,000 candidates. We call it high throughput assay. It's not that high throughput, but they would test 10,000 candidates to see a certain signal that if blue turns green, it's a good candidate or whatever it is, right? AI is making it possible to do it on screen and not having to do it as experiments, right? So something that would take months to identify. There's a professor here, Drew Adams, who does drug discovery. We can probably identify it in days, weeks at most. So, that context of AI is a great use, right? It's not regulated yet, but it's a great use. Clinical trial design. So this 2-3 decades of artificial blood clinical trials that ultimately failed and whatnot, there is a ton of data from all of those trials, right? And so AI is now while we are making the technology, we are using AI, we are part of some of the endeavors, going back to the clinical trial data and looking for signatures that are safety signatures or risk signatures. It would take, 1 billion people to do it manually, but AI can do it in a day, right? So while these kinds of things are emerging as what we call good use, we are probably all very aware of the bad uses in the current political environment, right? I don't have to talk about it, but we can all identify things that have happened or are happening as we speak that we know are not good uses of AI. But until a significant volume of pluses and minuses emerge, there aren't congressional efforts. This would require political efforts to put down regulations, right? That has not yet happened. So I'm sure it will happen, but it will take some time. And in fact, if our artificial blood becomes successful in clinical trials, because there is no precedent of a product like this to be used in patients, there is no reimbursement code for this. You can't bill a patient to give artificial blood. So another example where Congress has to come together and pass a bill for a reimbursement code that would allow insurance companies to pay for this.
Oh my goodness.
Right? So it's a complicated path. I'm sure we'll get there, but it's not obvious yet.
Yeah. Well, on that note, we're going to have to end, so we have time for the next panel. But will you join me in thanking our wonderful panelists?
Thank you so much. I appreciate it quite a bit. And yeah, you can email me anytime. I'm pretty good with responding to my emails. That's my decompression zone at midnight. So feel free to ask questions that you didn't get to ask.
And I wanted to just say the FDA was established in 1902. I was just doing some fact checking. 1906. And we had Tuskegee and stuff around 1932.
Yep.
And then we had clinical trials finally become the gold standard in the 1980s. So I think that
long path.
Timeline of what all we go through in order to get a regulatory body and then a process and then that becomes our standard is actually much longer than we may perceive. And so if AI is following suit, I think
maybe AI will make it shorter.
We'll see. Yeah, because the tech itself.
To be determined.
It's TBD.
Yeah,
the first iPhone, I think I heard you talking about how powerful your phone is. The first iPhone was 2007.
Oh my.
And this is 2025. And I can, control a satellite through my iPhone. So